Hello, everyone. My name is Kun Huang. I'm a professor in the Department of Medicine. Today, what we are going to discuss is an important topic in bioinformatics. It's called enrichment analysis. So the enrichment analysis is really a way for us trying to understand what's important or what are the common things among a list of genes. So where do I have the gene list? For example, if you do a comparative study, you generated RNA sequencing data for 5 Alzheimer mice, and you compared that with 4 control mice, and you got a list of differentially expressed genes. Then the question is, what's next? Usually the question comes down to what are the common things among those genes? Of course, besides those differentially expressed genes, sometimes a list of genes can be obtained from clustering of genes. You can get genes whose expression profiles are very similar across different samples. Or maybe you got genes that are homologs across a species or among many species. So there are many ways you can get a gene list. But many times the question is, what are the common things about the genes? What are the commonalities among the genes? For example, are they all involved, or most of the genes in your list are involved in cell cycle process, or maybe involved in immune response, or maybe involved in the NF-kappa beta pathway, or they may commonly interact with some gene, or they may commonly be regulated by a transcription factor or a set of transcription factors. Or maybe they are all known to be involved related to some diseases or abnormalities. Or maybe in many cases, especially in cancers, maybe those genes are on the same chromosome region. That's essentially, there may be a common copy number variance or something related to them to show the same behavior. But many times what you need to do is to identify the commonalities among the genes so that you can move to the next step to generate a new hypothesis. For example, if you found out that your genes are commonly regulated by a transcription factor, then it is very likely that you need to look into that transcription factor in your next study. So how do we find the commonality? So here we specifically listed in red, there are 3 categories of words that we look for, which are called molecular functions, biological processes, and the cellular components. And for those, they all belong to one thing, what we call the gene ontology. So one thing I would like to mention, when we want to find a commonality, that means we want to check with existing knowledge. By those existing knowledge, that could mean some database, or literature, or some other tables or lists that you curated. So this could be anything that you or colleagues or scientists or the community curated. So gene ontology is one set of such words, but organized in a specific way. So first we'll give an introduction of gene ontology because this is something you will see mostly commonly see in the report from the enrichment analysis. Gene ontology is actually developed by a committee. There is a specific international committee that they will vote on different words, different vocabularies related to biological processes, molecular functions, and cellular components. Biological processes means the ones such as mitosis, innate immune response, cell cycle. So many times, those are what we care most in biomedical research. Want to say, oh, those genes are all related to, cell cycle. are all related to genome stability. Molecular functions are the ones, for example, if this gene codes a protein that is a kinase or is ion channel or is some kind of enzyme. So those are the molecular functions. Those are not the so-called biological processes. Sometimes this is confused with biological processes because sometimes what you may want to be looking for is so-called biological functions. But actually in gene ontology, there is no such thing of biological functions. It's either biological processes or molecular functions. Then the third category of words is the cellular components. That is to say, for the protein product of this gene, where will the protein product be? Will it be on the membrane? Will it be on the extracellular matrix? Will it be in the nucleus? So that will be labeled and annotated. As you can see, we use the word annotation. That means someone will eventually manually do this. And not only they will manually do this, they will manually do this based on some evidence. This evidence could be really published papers, but sometimes could be a computational inference. So they will all be labeled if you actually look into the term, so-called ontology term for every gene. So if you go in there, if you're interested and you want to look for those ontologies, so this is how the ontology is organized, gene ontology is organized. And one thing you may notice is here we say there is more than 1.4 million gene products here. We all know human, we don't have those many genes. The reason is that this includes not only human genes, but all the genes that we know. And people try to annotate. It's not to say we can annotate every gene, but people try. So far, more than 1.4 million genes has been annotated. And that is why on the right side, you see the three categories also with each with more than 1 million annotations, gene products related to those annotations. So what actually you do if you click on the organism, and there are going to be a whole list of organisms that you can choose. And if you choose, for example, in this case, human, and it will tell you that now what you see is filtered for human only. And now what you see is the gene product is only 42,000. And for each category, they are also all less than 40,000. What do they look like? This is, for example, biological process. If you click on it, you'll have a whole large list. I mean, this is not a complete list, apparently. And if you scroll down, there is one of the component is called, one of the word is called cellular processes. And under the cellular process, there is one word called cell cycle. And there is a cell cycle, and there is a phrase called mitotic cell cycle. And behind each phrase, there is a number. That number indicates how many genes or gene products has been annotated with this phrase. So in this case, there is 1,095 genes that are related to mitotic cell cycle. And under mitotic cell cycle, 904 genes are related to mitotic cell cycle process. And under that, there are 14 genes are related to attachment of mitotic spindle microtubules to kinetic core. And under that, you can keep going and going. And clearly you can see this is a hierarchical structure. And the way they do that is, now if I have one gene specifically related to, say, mitotic sister chromatid bell orientation, by orientation. Then, based on this tree, based on this hierarchy, we automatically know it's related to cell cycle and it's part of a cellular process and it's part of biological process. So if you want to know more about each phrase, for example, mitotic cell cycle process, you can click on it. A menu will pop up. If you keep clicking on this mitotic cell cycle process, and this is what you're going to get. And the same thing on the left side, you can filter it by human, by Homo sapien, for example, or mice, whatever you want. But one function actually that might be used for some time is a download function. Here you can actually download all the human genes or the mouse genes or the rat genes that are labeled with this process. Sometimes people come to me and say, I want a list of all the immune response genes. This is one way actually you can do it by yourself. What you can do is then you can select the download button. It will give you a menu. The menu basically is not to say just will give you the genes or so-called gene names, but it will also give you its attributes, what ontology terms it's actually associated with, and where does those ontology terms come from. But if you really want to download, one thing you need to be sure is to select on this, drag it, click on this term, drag it to the right side, because otherwise it will not show up in your table. Once you select the selected fields, at the bottom, which I cannot show here, there is a button called download. And you download as a file. It's a text file, but you can actually open it in Excel. When you open it in Excel, it will look like this. Right now, I'm in this case, I actually put this gene product bioentity label on the top of the right list. And that is why you can see the gene symbols like CDT1 on the left, on the most left column. If you don't do that, you don't see that. And what you only see is a whole bunch of everything on the right side. That will actually become very awkward because you won't be able to know what actually, it will be very hard to actually know what's the name of the gene, what's the symbol of the gene. Please notice here in this table, the column C, it actually spells out the full name of the gene. For example, for CDT1, it's like DNA replication factor CDT something. Please note, officially, the column C is called gene name. Column A is not called gene name. Column A instead is actually called gene symbol. This is something also being often confused among researchers. So this is a quick introduction of the gene ontology. But besides gene ontology, those terms, you may say, I'm interested to know which pathway my genes are. That means we need some pathway database. One of the commonly used pathway database is CAG, this Kyoto Encyclopedia of Genome pathway database. And as you can see, if you go to CAG website, and this is what you see a whole bunch of things. But on the top, there is, in the middle on the top, there is the so-called KEGG pathway, CAG pathway. Say, okay, I want my database, pathways. In fact, when you get in here, what you see is there are a whole bunch of types of pathways. It could be a metabolism pathway. It could be a genetic information processing pathway or some disease pathways. So it's not organized as a typical what we call signaling pathway. It's kind of embedded in those kind of genetic information processing pathways, cellular processes pathways, human disease pathways, drug development pathways, and so on. So in that case, you have to really either search or go to browse. And the pathway is not only here, you can download the list of genes, but you can also look at actually as a pathway, how they are organized. And in here, example, metabolism pathway for tryptophan. And you can see those kind of 4-number the annotation for those enzymes, right, based on international standards. But you can also sometimes see disease pathway, like in this case, it's a colorectal cancer pathway based on known literatures. So you got things like KRAS is important, you got APC is there, and you got other important colon cancer related genes and how they are organized. This is based on known literature. KEGG is one of the databases we use. The challenge with KEGG nowadays is KEGG used to be an open source free database. Since a few years ago, they started to charge. If you really want to download the whole thing, you have to pay. There is an annual subscription fee. So people start to resort to other databases. Another database commonly used pathway database is called the Reactome pathway database. And I'm not going into details, and you can go there to browse by yourself. And also sometimes there's the one that's really not free, for example, Ingenuity pathway analysis. And we have another session specifically about Ingenuity pathway analysis. Sometimes you may ask, are there any other pathway databases? There are many. There is actually a website listing all the pathway or most of the pathway databases called pathguide.org. On the left side, you can see there are many different pathways we are talking about. How many pathway databases it collected? 702 so far. It's still increasing. The first time I saw this website was about 12 years ago, and at that time there was only about 100 databases. Now there are more than 700 databases here already. So you can have all the databases. Now the question is, how do we actually do the enrichment analysis? Remember, as we mentioned, the enrichment is based on prior knowledge. For example, if you have a list of genes, maybe 50 of them, and some of them you noticed, maybe more than commonly, there seem to be quite a few genes related to cell cycle genes. So one thing you can do is you can get the list of all the annotated cell cycle genes, and you look at how common they are in the background of the entire genome, and how significant is this commonality between your gene list and the annotated cell cycle genes, or annotated immune response genes, or annotated metabolism genes, right? So there has to be some way, mathematically or statistically, that we can evaluate how significant is this intersection, so that you know what you observed is not something random. It's likely to be have a biological meaning. So the way they do that is actually through some mathematics. Right? Think about this. Now you have N genes in a background of capital N genes of the entire genome, or some specific gene list you selected. Your gene list has lowercase n genes, and for a specifically annotated gene set, for example, cell cycle genes, there are M of them. So in your gene list, there are K genes shared in common with this annotated gene list. So what's the probability that you will observe this? If you remember your high school math, this is essentially to say, okay, if I divide my N genes into the ones that are in this green circle, which are K genes, and the ones that are outside the green circle, which are N minus K genes, what's the chance that I'm going to see that? So first, within the green, the chance you're going to see K genes is M choose K. And outside the green, the chance you're going to see those N minus K genes is N minus M choose N minus K. But in total, there are how many ways you can choose n genes from this entire genome background? It's n choose n. So this is essentially the so-called hypergeometric distribution. And that's essentially the p-value that you can see, observe exactly k genes has that annotation. But this may not be exactly what we know. Sometimes it could be even more of those. It's just because we don't know those genes yet. So you need to consider other even more extreme cases. So the Fisher exact test is used to test this. It's actually just a sum, just add all the probabilities for similar contingency tables with equal or even smaller probabilities. And then they put together, that become the p-value for this Fisher exact test. That is essentially how likely you are, if this is random, right? How likely you are going to see this, what you observed, or even something even more extreme than what you observed. So just bear in mind, if you don't really feel comfortable with this math, that's fine. But just bear in mind, this p-value just give you a metric of how likely you are going to see this as a random event. So if it's very small, that means it's very unlikely to be a random event. And then the biological meaning might make more sense in this case. So surely we are not going to ask you to do this for every term, gene ontology term or possible database. There are many softwares out there that can allow you to do this. In a later session, Dr. Li Chen will talk about another tool called David. And I'm going to start actually by demonstrate a simpler tool called TopGene. TopGene is very easy to use. And even though the functionality is different with David, but it has the basic functions of the enrichment and it's very straightforward and intuitive to use. Of course, there are other tools like GC, and for the one that are commercial like Metacore or Ingenuity Pathway, and many others. So here are the websites, and we are going to specifically using 2 gene lists to demonstrate how to use TopGene. And here is the screen capture for TopGene. In the TopGene website, TopGene is developed by Cincinnati Children's Hospital, and there are multiple functions. We are only going to focus on this so-called TopFun function, that is functional enrichment. Okay, we'll switch to another video and for the demo of TopFun.